going to be on diseases that occur due to amyloid. And we'll have a Socrative room for today. And if you guys would like to log in, the room code is Mohammed, MOHAMED 2667. So I'll give you guys a couple seconds to log into that. And I'll put it in the chat as well, so you guys can refer to it later if you forget. Okay, so here's the syllabus, and it pretty much outlines what I'm going to talk to you guys about today. So first of all, we're going to go through what amyloid actually is and how it goes about causing disease. And then we're going to illustrate it with a few examples of different neurological diseases, as well as the diseases of some of the other systems as well. So there'll be quite a lot of clinical relevance today. And you'll see that I've name dropped some experiments that you guys can use in your essays if you're interested in experimental evidence and writing essays for this topic. For the most part, I'm not actually going to explain these experiments because it's all considered extra material. And in this lecture, I kind of want to talk about the core stuff. But in case anyone's interested, then the experiments are going to be there. So first of all, what is amyloid diseases and why do they matter? So the key underlying pathology in amyloid diseases is protein misfolding and aggregation. And this is associated with impairment of tissue and cell function and ultimately degeneration. And different amyloid conditions have different misfolded protein. They have different sites of deposition of this misfolded protein. They have different causative and risk factors. And as a result, the clinical consequences are different as well. And often we think about neurodegenerative diseases of aging, which are Alzheimer's and Parkinson's most commonly. But there are also some systemic amyloidoses that affect other organs and other organ systems. And there are some infectious variants as well. And these infectious types are quite rare, but they are fatal. So why is it important to learn about amyloid? Well, the cases of amyloid diseases as a whole are rising rapidly, and we expect that in the next 30 years, there'll be more than 2 million cases of Alzheimer's in the UK alone. So these diseases cause a lot of morbidity and mortality, so it's important to know about them, especially as we have an aging population. And at the moment, we have no cures for amyloid diseases, and all we can do is alleviate symptoms. So this is partly due to the fact that by the time the symptoms have appeared, the degeneration has progressed so far that we actually can't reserve it or modify the diseases. So it's important to learn a bit about that as well. So for the first Socrative question, fill in your answers. And the question is, which protein structure causes dysfunction and cell death in amyloid disease? Okay, so the most common answer is the misfolded protein monomers, which is a very rational answer, and I understand, because I've just told you guys that it's the misfolding that causes disease in amyloid diseases. And some people have spoken about the fibrils and the aggregates of the fibrils as well, which is also a key part of amyloid pathogenesis. But as I'll explain to you guys today, the part of the protein that actually causes the cell death is the oligomers. And we're going to go through how that happens just now. So essentially with amyloid disease, if you understand this slide and the next slide, you basically understand all of amyloid. And essentially what you find in amyloid diseases is that you have this normal host protein, which usually has an alpha helix rich structure. This normal host protein misfolds from the alpha helix rich to the beta sheet rich structure. And various factors contribute to this misfolding, and that includes proteolysis, the naive fold of the protein being a bit unstable anyway, various denaturing conditions, etc. Anyway, so it folds from alpha helix rich to beta sheet rich. And these misfolded monomers should usually be cleared as they're formed, but there are various factors that result in them persisting. And when they persist, they ultimately cause disease. So for example, aging just advanced age, can cause the clearance mechanisms to be impaired. The protein itself can be produced excessively, for example, in a chronic inflammatory disease. The protein could have a greater propensity to be misfolded. For example, if there's a genetic predisposition, or the misfolding may actively be propagated in the example of prion diseases, which we'll go through later. So these misfolded monomers then form misfolded oligomers, and the oligomers are the key mediators of cellular toxicity. And the oligomers can also form fibrils later down the line as they continue to get longer. But it's important to note that the significance of the fibrils is kind of disputed. In part, they may be protective because it's a mechanism to remove the toxic oligomers, but they could possibly cause physical mechanical disruption to the extracellular matrix. And they can also become protease resistant, so they can't be cleared normally. But the evidence shows that the oligomers are the ones that cause the cell death rather than any other part of the protein structures. So the key takeaway is oligomers are the primary mediators of cell death in amyloid disease. Okay, so how do the oligomers cause cell death? So one of the proposed mechanisms is calcium dysregulation. So the oligomers form these membrane pores which cause the calcium to be unbalanced. And then calpain, phospholipases, endonucleases, etc., are activated, which results in necrosis, and caspases are activated, which results in apoptosis. So there are multiple different mechanisms of cell death. And another one is excitotoxicity as well. Mitochondrial dysfunction is also another mechanism, and the oligomers basically cause pores to open in the mitochondrial membrane, resulting in the release of reoxide oxidant species and apoptotic factors as well. And some other key mechanisms are lysosomal dysfunction, which results in impaired autophagy and therefore impaired clearance mechanisms too. And the oligomers also alter the signaling throughout the cell too. So essentially, those are the key ways that the oligomers cause cell dysfunction and death. And I've put a couple of studies in here that demonstrate oligomer toxicity, and you can look through these in your own time if you're interested. So just a quick slide to show the key misfolded proteins in some key amyloid diseases. So in Alzheimer's, the protein that misfolds is derived from APP, amyloid precursor protein, and it's called beta amyloid. In Parkinson's, it's called alpha-synuclein. In the various prion diseases, such as variant CJD and fatal familial insomnia, it's prion protein, PRPSC, and that's derived from PRPC. And then in secondary amyloidosis, it's serum amyloid APP, and in type 2 diabetes, which is also interestingly an amyloid disease, it's the islet amyloid precursor polypeptide. As you can tell, amyloid scientists love lots of really long words. So these proteins all undergo the alpha to beta transformation that we mentioned, and they happen in different parts of the body which are relevant to each specific disease, causing damage and cell death. And we'll go through how exactly that happens in just a second. So first of all, let's look at the diseases of aging. And the first disease to talk about is Alzheimer's. And this is a common disease, and it's the most common cause of dementia. So Alzheimer's is usually sporadic, and it's caused by a combination of genetic and environmental factors. And there are also some rare early onset forms that are more severe and that occur even as young as 20s or even 30s. And these are caused by inherited mutations in amyloid precursor protein, APP, and presenilin 1 and 2. And these early onset forms are significant because they can give us insights into some of the pathophysiology of the sporadic, normal, regular form of Alzheimer's. So in terms of how Alzheimer's affects people clinically, it's a pretty difficult disease, and it has major neurological, psychiatric, systemic, and social impairments. And it usually starts off with some cognitive impairment and some short-term memory loss, and then ends years down the line with patients being totally dependent on care and or shortening their lifespan. So as we mentioned before, the key pathological protein in Alzheimer's is derived from APP, which is normally just a large membrane glycoprotein, which is found just throughout the brain. APP is typically cleaved by two proteases called beta and gamma secretase. And this cleavage produces a protein fragment called beta amyloid. And beta amyloid undergoes this alpha to beta oligomer conversion, and that results in toxicity via the mechanisms that we mentioned earlier. So the permeability, the calcium dysregulation, the mitochondrial dysfunction, etc. And in addition, extracellular beta amyloid plaques are formed, and that triggers a local chronic inflammation too by the glial cells in the brain. So as well as beta amyloid, another protein that is kind of acknowledged in Alzheimer's is called tau. And I mentioned this primarily just because you may have heard of it or you may come across it in your reading. So I just thought I'd tell you guys about it. So tau is a microtubule associated protein, and it's involved with intracellular trafficking. And in Alzheimer's, tau becomes hyperphosphorylated, and it forms these fibrils as well, which aggregate intracellularly in structures called neurofibrillary tangles. And these intracellular inclusions differ from the typical amyloid plaques, which are extracellular inclusions. And it's not quite clear what the NFTs do, and if they're harmful, if they're protective, because some people think that it might be a protective mechanism in response to beta amyloid toxicity. But anyway, I say all that to say that the key amyloid protein in Alzheimer's is beta amyloid. So ultimately, these beta amyloid oligomers result in the progressive, irreversible neuronal loss, and that neuronal loss starts off subcortically and then becomes cortical as well. And it particularly affects the cholinergic neurons of the basal forebrain. And over time, you can see that the cortex shrinks if you look at either imaging of someone with Alzheimer's who's had it for quite a while, or even if you look at post-mortem brain samples as well. And you can see the sulci and the gyri are much deeper than they are in a normal human brain. Again, just a couple slides on a couple experiments on Alzheimer's. So now let's get back to Socrative and look at the next question. And the next question is, which of these drugs has shown capacity to slow the progression of Alzheimer's? I don't really expect you guys to know this, by the way, but we'll go through it in a sec. Okay, so most of you guys have answered now. Okay, so anticholinesterase seems to be the most common question. I see you, last person who just clicked the right answer. And that is definitely the first line drug that we use in Alzheimer's. But the problem with it is that it only relieves symptoms, but it doesn't slow the progression of the disease, which is quite a shame because, as you can imagine, it means that people still have worsening neurodegeneration and worsening symptoms over time. So in terms of the therapeutic approaches to Alzheimer's, at the moment, we don't have a cure for it. So the anticholinesterases, as you guys correctly recognize, are a common drug used to treat Alzheimer's. And these include drugs like rivastigmine and donepezil and so on. And what they do is they increase acetylcholine availability in the brain because the cholinergic neurons are among the key neurons killed. So basically, they increase the availability of acetylcholine to try to mitigate the effects of Alzheimer's. But this doesn't change the course of the disease because it doesn't target the underlying pathology. So whilst it may relieve symptoms, we would like to use an intervention that targets the underlying pathology. Another key approach used in Alzheimer's is NMDA receptor antagonists, and these drugs that include memantine work by inhibiting a neurotransmitter called glutamate, and that effectively is thought to decrease excitotoxicity and to decrease that mechanism of cell death. And it's actually been shown that it can modestly slow down disease progression as well. But even so, that doesn't work in everyone, and it's only a modest decrease in disease progression. So like I mentioned, ideally, we don't just want to relieve the symptoms, we want to tackle the underlying pathology, the underlying amyloid deposition. And because of that, there have been some attempts to develop beta and gamma secretase inhibitors. And if you remember, the beta and gamma secretase enzymes are those which cleave that APP to produce the beta amyloid in the first place. So in theory, that should slow down the disease progression, but sadly, in practice, they've actually failed all the phase 3 clinical trials so far. And the EPOC trial was actually a recent trial that was terminated in 2017 because they found that there was no real clinical benefit, which is a shame. So we're still quite a way away from developing some proper treatments for Alzheimer's. And perhaps part of that is understanding the pathology a bit better. Okay, so that's a wrap on Alzheimer's, and now we're on to Parkinson's. So before we start, let's look at this question on Socrative. And the question is, neurons in which part of the brain are primarily lost in Parkinson's? And also I've realized that the question is worded wrong. I mean, which type of neurons are primarily lost in Parkinson's? Okay, most of you guys have answered. Fantastic, you guys did perfectly. So it is dopaminergic neurons. And if we go on to the next slide, so we'll talk a little bit about Parkinson's. And Parkinson's is the second most common neurodegenerative disease following Alzheimer's. And it affects more than 1% of over 60-year-olds, which is quite common. It primarily involves the progressive degeneration of the midbrain dopaminergic neurons. And these neurons go from the substantia nigra pars compacta to the striatum. And these neurons in this pathway are basically involved in some key motor pathways. So many of the recognized symptoms of Parkinson's are motor symptoms, and they include things like the tremor, the rigidity, the slowed movements, etc. But it's also important to remember that there are some non-motor effects of Parkinson's, and these include things like anxiety, cognitive impairment, sleep problems, and loss of the sense of smell, which can actually come on before the motor symptoms, but it's something to be aware of as well. And as with Alzheimer's, Parkinson's is usually sporadic, and it has some complex gene environment interactions, but there are also some early onset genetic forms. So what role does amyloid play in Parkinson's? So the key protein in Parkinson's is called alpha-synuclein. And alpha-synuclein is normally found in the presynaptic terminals in the brain, and it's thought to be involved in neurotransmitter release. So alpha-synuclein misfolds from the alpha helix rich to the beta sheet rich form, and aggregates in these structures called Lewy bodies. And again, we see the same pathological patterns of calcium dysregulation, lysosomal and mitochondrial dysfunction, etc. And ultimately that leads to impaired autophagy and oxidative stress. And then eventually we have neuronal death as well, as a decrease in astrocytes, so the support cells, and an increase in microglia, which are the inflammatory cells. And by the time the symptoms have appeared, and that's years and years down the line from the start of this pathology, more than half of the dopaminergic neurons are lost. So as you guys can imagine, it's quite difficult to mitigate these effects later down the line. And again, some studies. So considering that there's such extensive neuronal loss by the time that patients have presented to their doctors, it's quite difficult to do anything beyond symptom relief. L-DOPA is the first line treatment of Parkinson's. So it's a precursor of dopamine, which can be orally administered, and then it can readily cross the blood-brain barrier, whereas if you give dopamine, it won't cross the blood-brain barrier. So you give the precursor, crosses the BBB, and then when it gets to the brain, it's converted to dopamine to replace the endogenous dopamine that's lost. So it's kind of a similar theory to the anticholinesterase, in that it replaces the endogenous protein, the endogenous transmitters that are lost, and provides some symptomatic relief, but it's not without its drawbacks. So after several years of using L-DOPA, it can actually create its own motor defects. And once you have done that, it's even more resistant to treatment to treat these motor problems. So a more novel treatment is deep brain stimulation. And that basically involves you surgically implanting an electrode into the basal ganglia, which controls motor coordination in the brain. And this works really well in some patients, but not in all. But it's a pretty promising part of future development of Parkinson treatment. And there are also some early stage trials using induced pluripotent stem cells and a new assay that's being developed to try to diagnose Parkinson's earlier. It's not core material at all, so I won't go into this unless anyone's interested in the end, and the slides are here for reference anyway. Okay, so now we've spoken about Alzheimer's and Parkinson's, and those are two common diseases which typically affect older people. And in the next section, we're going to talk about prion diseases, which on the flip side are really, really rare, and they typically affect much younger people. So the definition of a prion is a small proteinaceous infectious particle that resists inactivation by procedures that modify nucleic acids. So in other words, unlike a bacteria or virus or other pathogen that depends on its genetic material to maintain its survival and its infectiousness, prions can survive, be transmitted, and cause disease purely in their protein form. So like other neurodegenerative diseases, prion diseases are progressive and they're irreversible. But unlike the others, they are invariably fatal. So whilst you can have Alzheimer's and Parkinson's, and you could not die from it because it takes quite a few years for it to progress to the point where it's fatal, if you have a prion, it will kill you and it will do it very, very quickly in even a matter of weeks. So prions can occur due to a sporadic genetic mutation, can occur due to a genetic mutation or inheritance of a mutant gene, or they can be caused by infection. Can you guys see the arrows on this slide? Because they seem to have disappeared for me.
Yeah, we can.
Okay, lovely, thank you. Okay, so the key amyloid protein in prion disease is very creatively called prion protein. And prion protein, PRP, is normally found in the alpha helix rich form. I'm sure you guys are sick of me saying the word alpha helix rich, but the alpha helix rich form is called PRPC, and it's found abundantly in the central nervous system. It's a type of membrane protein, and of course, we don't know what exactly it does, but it seems to be neuroprotective in some way. So anyway, you guys know the drill by now. PRPC undergoes the alpha to beta conversion, at which point we call it PRPSC. And its oligomers cause toxicity in all the ways that we've spoken about before. And they're also known to accumulate in the endoplasmic reticulum, which triggers the unfolded protein response and stops the translation of other proteins. And the interesting thing about PRPSC is that it also promotes the nearby normal PRPC to become PRPSC and creates this massive positive feedback loop, creating lots of abnormal prion protein. And all in all, it causes massive neurodegeneration. And if you look at some histology, which we definitely won't be doing today, you'll see really large acellular areas on the histology. And then in the later stages, you can actually just see chunks of brain missing, which is really horrible. So anyway, there are a few different prion diseases, and today we're just going to focus on variant CJD. So this is an infectious form of CJD, and CJD is a prion disease which is closely related to BSE, which is bovine spongiform encephalopathy. It's also known as mad cow disease, and CJD is thought to be contracted by eating meat from cows that have had this BSE infection. And the name BSE just gives you all the information you need. So bovine, it comes from cows. Spongiform is because the brains look like sponges because they just have punched out essentially holes in them afterwards. And encephalopathy because it causes behavior and personality changes. So anyway, CJD often affects younger people and it causes very rapid neurodegeneration. There's a broad spectrum of symptoms, and this includes rapidly progressing dementia. And the difference between delirium and dementia primarily being that delirium is transient and it's reversible, whereas dementia is progressive and it's irreversible. So it causes this dementia, these psychiatric symptoms, including hallucinations and personality changes, and various musculoskeletal effects as well, such as jerks and stiffness, to just name a few of the symptoms. And eventually, CJD proves fatal within a few months. As well as this infectious cause of CJD, known as variant CJD, there is a sporadic version, which is just caused by de novo mutation in the prion protein gene called PRNP, and a familial version caused by an inherited mutation, and an iatrogenic version, so a doctor-caused version, caused, for example, by transplanting a contaminated brain graft from an undiagnosed CJD patient. And on that very positive note, I've included a slide on another prion disease called fatal familial insomnia. It's right-hand side syllabus stuff, so I'm not going to go through it, but it's here if anyone wants to look at it. In terms of treating prions, at the moment, we can only provide symptomatic relief and end-of-life care. So there are some potential disease-modifying treatments that are in development, including some old antihistamines and an oligomer modulator. And they've shown some success in mouse models, but they're still a very long way from being on the market. And what we can also do is to passively immunize people who have been / might become exposed to prions. If, for example, there's an outbreak of mad cow disease nearby, but that requires us to identify them very early to be able to provide that kind of treatment. So back to Socrates. And the question is, which of these is not a cause of prion disease? Wonderful, most of you guys have answered now. Amazing, PRPC infection. So all of these others can cause prion disease, and PRPC, as you guys correctly remembered, is the normal version of PRP. And the Giromata graft is a type of brain graft that if the person that you're getting it from has CJD, then that could cause infection in the recipient. Okay, so we've spoken about what amyloid is and the diseases of Alzheimer's and Parkinson's, which are diseases of aging usually, and we've spoken about the rapidly fatal prion diseases. So in this last little chunk, we're going to talk about the systemic effects of amyloid in chronic disease. And it's a really, really small part of the syllabus, so I'm not going to go into too much depth, but essentially secondary amyloidosis refers to amyloid pathology, which is not caused primarily by amyloid deposition, and instead amyloid deposition occurs after or alongside the other primary disease process. So for example, in some parts of the country and some parts of the world where there is TB, chronic TB infection can cause also amyloid deposition. And unlike the diseases that we have already spoken about, secondary amyloidosis affects the whole body and not just the brain. So some of the key organs that are involved are the heart and the kidneys. And it can cause heart failure, it can cause various renal impairments, and it can cause a range of other symptoms as well. And another chronic disease for which there's increasing awareness about the role of amyloid in is type 2 diabetes. And here the key protein is IAPP, and that normally regulates various different aspects of energy homeostasis. And it becomes amyloidogenic via, of course, unclear mechanisms, and deposits in the pancreatic islets. And this is associated with inflammation, islet dysfunction, and death. And it seems to also be the cause of the failure of islet cell transplants, which sometimes is used as treatment for type 2 diabetes. And one last Socrates question is, which of these is not an established mechanism of cell death in amyloid disease? Okay, amazing. Fibril formation was the correct answer. There may be some role for it, but it seems that it's not the primary cause of cell death. And excitotoxicity can be a mechanism of cell death in amyloid disease. And I think the evidence that NMDA receptor antagonists work may also support that hypothesis as well. So that is the end of this talk. Thank you guys all for tuning in. I hope that was useful for you guys. And as usual, if you have any questions, please do either write them in the chat function or shout them out or send an email afterwards.